Assalamu alaikum dear students. Today we will study about the photosynthetic pigments. Photosynthetic pigments. A photosynthetic pigment is a biomolecule which has a remarkable property of absorption of light. यानी एक फोटोसिंथेटिक पिगमेंट जो होता है उसके पास एक सलाहियत होती है, वो सूरज की रोशनी को एब्जॉर्ब करता है. And this is a unique property associated with the pigments, photosynthetic pigments. Now, the photosynthetic pigments are present inside the thylakoid membranes of the chloroplasts, and they are basically meant for absorption of photons or light energy. They are broadly of two types. Number one type you call as chlorophylls, and number second type you call as carotenoids. Chlorophylls are green in color, and they are the main photosynthetic pigments. Why the chlorophylls are green? This is because they absorb the light from both ends of the visible spectrum, and they reflect most of the green wavelength. This is the reason why they appear green to us. And on the other hand, the carotenoids are colored variously. For example, they can be orange, they can be red, they can be yellowish to brown. And carotenoids are the helping pigments, or you can call them as accessory pigments. However, they are very, very important. They are not less important. So these two make the photosynthetic pigments. Now let us first take up the chlorophylls. I told you that the chlorophylls are green in color and they are the main pigments in the photosynthesis. They have many types. For example, we have chlorophyll A, B, C, D, E. Out of these we have the two, that is chlorophyll A and chlorophyll B. These are present in the higher plants. So let us take into consideration these two types of photosynthetic pigments or chlorophylls. Chlorophyll A is bluish green, blue-green in color, and it is more soluble in petroleum ether. Petroleum ether and organic solvent. इसमें ये ज़्यादा soluble होता है. It has an empirical formula C55H72O5N4 Mg. And this chlorophyll A molecule, the chlorophyll A molecule is said to be universal chlorophyll molecule. This is because it is found in any plant group, whether the plant group belongs to lower plants or higher plants, it should contain chlorophyll A. That is why it is called as universal chlorophyll molecule. Moreover, it has a significance regarding the choice of photosynthetic pigments out of this. I told you previously that there are two pigment systems, pigment system first and pigment system second. The reaction center of any pigment system is chosen from chlorophyll A only. So that way it is very important. Now come to chlorophyll B. Chlorophyll B is olive green in coloration, and it is more soluble in methyl alcohol. मिथाइल अल्कोहल, इसमें ज़्यादा सॉल्युबल होता है in comparison to in comparison to its solubility in other organic solvents. So chlorophyll B, it has an empirical formula of C55 H72 O6N4 Mg. This is the structural formula of chlorophyll B. So this was about the chlorophylls. Chlorophylls are found in the green parts, and where it is in abundance, it will impart the green coloration. After chlorophylls, the other type is carotenoids. Carotenoids. As I told you that these are accessory or helping pigments, and these are further of two types. One type you call as carotenes, and the other type you call as xanthophylls. First of all, we will take the carotenes. Carotenes have a general formula C40 H56. This is the empirical formula for carotenes. So carotenes have carbon, hydrogen, but they lack oxygen. And carotenes are orange to red in coloration. On the other hand, we have xanthophylls. Xanthophylls are represented by the formula C40 H56 OX, and here X will be any number from 1 to 8. That is, oxygen atoms can be either 1 or any number up to 8. So these xanthophylls are oxygenated carotenes. Oxygenated carotenes, or these are also called as carotenols. Carotenols इनका दूसरा नाम है. So these are carotenoids. The carotenoids absorb the wavelengths from one end of the visible spectrum, that is from the blue-violet region, and they reflect the light on the other end, that is towards the orange-red region. That is why they appear colored to us variously other than green. So these are the photosynthetic pigments. Now if you see the examples, we have carotenes like alpha carotene, we have beta carotene, we have gamma carotene, different types of carotenes. Out of these, beta carotene is most abundantly found in the plants, and in our body this beta carotene is changed into vitamin A. On the other hand, the examples are various types of xanthophylls include the zeaxanthin and violaxanthin. So these are examples of xanthophylls. So the various types of photosynthetic pigments, these are organized in the form of photosystems or pigment systems in the thylakoid membranes, and their job is to harvest maximum possible quanta of light energy to be utilized in the process of photosynthesis. Now, we have a phenomenon that you call as fluorescence, and another is phosphorescence. Fluorescence, and another phenomenon you can call as phosphorescence. When you deal with the photosynthetic pigments, you may come across these terms, that is fluorescence and phosphorescence. Now let us see what is fluorescence. Fluorescence is actually immediate emission of long wave radiation while the electron is returning from first excited singlet state to the ground state. This is the definition of fluorescence. On the other hand, the definition of phosphorescence is the delayed emission of radiation, long wave radiation, while the electron is returning from triplet state to the ground state. So this is the difference between fluorescence and phosphorescence. I will repeat the definitions. This fluorescence is the immediate emission of long wave radiation while the electron is returning from first excited singlet state to the ground state. On the other hand, the phosphorescence is delayed emission of long wave radiation while the electron is coming from triplet state to the ground state. Now we will understand what the fluorescence means actually and what is the mechanism by which the fluorescence occurs in pigments. For that purpose, we will take a pigment solution, especially the chlorophyll solution. Chlorophylls and carotenoids, these are dissolved, these are soluble in organic solvents. So you have to take any solvent, organic solvent like petroleum, ether, acetone, etc. And you can dissolve the pigments in them to obtain a solution called as pigment solution. Now this pigment solution will be illuminated to light. Suppose this light is the incident light. Incident light falling upon this solution contained within the test tube. Some of the light will be reflected. So this will be called as reflected light. Reflected light. And some light will be transmitted. It will come through this solution and this will be called as transmitted light. Transmitted light. If you see this solution in the transmitted light, it will appear normally green to you. It will be green in color. On the other hand, if you see this solution in the reflected light, it will appear red in color. Red in color. So this paradoxical situation that arises here, that is the solution simultaneously appears red and green when viewed either through reflected light or transmitted light. This is because of fluorescence. And fluorescence can be understood when we take the energy status of the electrons in the constituent atoms of chlorophyll molecule. Now let us take into consideration the ground state of molecule S0. And then there is a higher energy state that you call as S1, first excited singlet state. There is still another that you call as S2 state, second excited singlet state. Suppose a chlorophyll molecule has 2 electrons in the ground state before it has absorbed any photon. And these 2 electrons which are in S0, they have opposite spins. That is, one has clockwise spin and other has anti-clockwise spin. Suppose a red photon is being absorbed. Red photon. Red photon is not much more energetic. It has least amount of energy in comparison to other photons that may be belonging to various other wavelengths of the visible spectrum. So, red photon is absorbed. It will excite the electron to S1. So, the electron will be excited to this, but the spin will remain same. if this electron is excited, it had a clockwise spin. The spin will remain same, no change in the spin. However, the electron is excited to S1, first excited singlet state. Here this will remain for a period of 10 raised to power minus 12 seconds. 10 raised to power minus 12 seconds के लिए ये रहेगा यहाँ पे, क्योंकि this is the half life period of this first excited singlet state. If an electron acceptor is present at S1, the excited electron will be captured by the electron acceptor, and we will say the energy of the electron or energy of the photon has been fixed into the reactions of photosynthesis. Otherwise, if that suitable electron acceptor is not available there, the electron will return to the ground state within a span of 10 raised to power minus 12 seconds, losing the previously absorbed energy in the form of long wave radiation, that is red. ये रेड लाइट की सूरत में अपनी एनर्जी को खारिज करेगा और ग्राउंड स्टेट में चला जाएगा. एंड दिस immediate emission of long wave radiation while the electron is coming from S1 to S0 is called as fluorescence. अब इस पिगमेंट ने क्या किया? इसने फ्लोरेसेंस को एंश्योर किया. ये फ्लोरेसेंस इसलिए हुआ क्योंकि इलेक्ट्रॉन की जो एनर्जी थी वो फोटोसिंथेटिक प्रोसेस में यूटिलाइज नहीं हो गई. चलिए ठीक है. अगर हम यहां पे क्या करते हैं, ये जो इलेक्ट्रॉन है ये चला गया S1 के अंदर. सपोज रेड फोटोन वाज absorbed, and the electron was excited to S1, जैसे ये हमने दिखा दिया. Sometimes what happens if the electron acceptor here at the S1 is not available, and this electron tries to reach the S0 by losing the energy associated with it. But sometimes it cannot lose the whole amount of energy associated with it in one go. ये एक ही लम्हे में इस सारी एनर्जी को लूज नहीं कर पाता है कभी कभार. तो क्या होता है? द इलेक्ट्रॉन कम्स डाउन एंड it will come down losing only a small amount of energy associated with it that will come out in the form of heat. So there will be heat loss, and because of this the electron reverses its spin. इसके पास जो स्पिन था यहां और यहां S1 और S0 में वो था clockwise spin. Now the spin becomes changed, it becomes reversed, and it is converted into anti-clockwise spin. So, if this electron with anti-clockwise spin returns to the S0, it has here this counterpart that too has an anti-clockwise spin. So the spins will be parallel, and these cannot be accommodated here because no two electrons can be present in the same energy level that have same spins or parallel spins. So what will happen? ये जो इलेक्ट्रॉन यहां पे है ये लगता है कि फंस गया. The electron seems to be trapped in a slightly lower energy level than S1, that is called as T state or triplet state. This is called as triplet state. अगर इलेक्ट्रॉन ने ये ट्रिपलेट स्टेट क्रिएट किया, this triplet state has a half life period of 10 raised to power minus 9 seconds. So for a longer duration the electron will be trapped there, but within a span of 10 raised power minus 9 seconds the electron will come to the ground state, losing the previously absorbed energy again in the form of long wave radiation, that is red. And this phenomenon is the delayed emission of radiation while the electron is coming from triplet state to the ground state, and this is called as phosphorescence. इस phenomenon को आप क्या बोलोगे? इसको आप बोलोगे phosphorescence. Phosphorescence. So phosphorescence is the delayed emission of long wave radiation while the electron is coming from triplet state to ground state. ये जो यहाँ पे आएगा, जो इसका स्पिन इसके पैरेलल हो गया था, दैट इज इसका भी क्या हो गया था, एंटी क्लॉकवाइज, ये फिर से क्लॉकवाइज हो जाएगा और नीचे आ जाएगा. ओके, सो दिस वाज द डिफरेंस बिटवीन phosphorescence and fluorescence. Now I have to tell you one more point. We say that in the process of photosynthesis, red light is very efficient, or rather most efficient type of wavelength in the photosynthetic process is the red wavelength. So we must be in a position to know how red light is most efficient wavelength in the process of photosynthesis. For that purpose, we will say here, suppose we have two electrons again here, one has the anti-clockwise spin and another has, this has clockwise spin and this one has anti-clockwise spin. चलिए ठीक है, अब क्या करते हैं हम, this S1 state that you call as first excited singlet state. This is very important. This is because whenever any electron is excited and it is emitted by means of reaction center, P680 or P700 reaction center, the electron goes to S1 and the S1 energy level contains, or we can say there is an electron acceptor that has just the energy which is associated with S1. So there is always present an electron acceptor so that the electron can be accepted by that substance and that substance will be reduced and this will generate a photosynthetic electron transport chain. The electron acceptor has never the higher energy, that is, it never exists in this S2 or second excited singlet state. So always in the process of photosynthesis, it is the first excited singlet state energy which fixes the photosynthetic reaction, photosynthetic energy in the process. So electron acceptor that you call as primary acceptor of electrons, that is worth the energy that is present in S1. Okay, so if red photon is absorbed, it directly excites the electron to S1 without any heat loss, without any wastage of energy. The red photon promotes the electron to S1 where electron acceptor can accept it. Now consider the blue photon or any other higher energy photon. Blue light is very much energetic. It has a shorter wavelength but a higher amount of energy per quantum. Blue photon, if absorbed by the electron goes to S2, second excited singlet state. The spin will remain same, no change in the spin. But here this electron won't be accepted by means of any primary acceptor. So this electron has to first create the S1. How it can create S1? It can create S1 energy level by heat loss. So the extra energy that was contained in a blue photon was here wasted because at S2 state the electron was not accepted by any primary electron acceptor. It has to necessarily create S1 in order to be accepted by the electron acceptor. So heat loss occurred. And when it is able to create S1, only then it can be in a position to participate in the process of photosynthesis. So we say that the red light or red wavelength is efficient in the process of photosynthesis, rather most efficient form of wavelength in the process of photosynthesis. These other wavelengths which have lower values of wavelengths but higher energy per quanta, they are effective but not as efficient as red light. I hope you have understood this phenomenon. So this was about the phosphorescence and fluorescence and the reason why the red light is most efficient light in the photosynthesis. We will return after a few seconds.